Okay. Hello, everyone. Thank you so much for coming today. Thank you so much for being here. My name is Julie, and along with Ash, who is here, we are the Walk-in Engagement Associates. On behalf of Brain Tumor Foundation of Canada, we would like to extend our gratitude and a huge thank you for your ongoing support and dedication. Because of you, we are one step closer to finding a cure. I'm going to introduce our team today and who will be conducting the lab tour. Jessica de Santis is a fourth-year PhD candidate from Brazil, working as a visiting researcher at the BC Cancer Research Centre in Vancouver, under the supervision of Dr. Alberto Delaidelli and Professor Poul Sorensen. And we have her colleague Ferris here as well. Their research focuses on novel therapeutics approaches for pediatric medulloblastoma, and they have recently received an award from Brain Tumor Foundation of Canada for their project entitled Medulloblastoma Plasma Membrane Proteomics to Inform Optimal Immunotherapy Design. Their work involves the study of proteins exposed on the membrane of brain cancer cells to identify new targets for immunotherapy, such as CAR T cells. On another project, they are working on the metabolism of high-grade medulloblastoma cells to understand the function of a gene associated with patients' poor prognosis. Both projects aim to improve the patient's limited therapeutic approaches available. Jessica is a biomedical scientist with background in clinical pathology and immunogenetics, and completed her master's in genetics at the University of Sao Paulo. Her PhD project is focused on pediatric brain tumors and characterizing therapeutic targets to enhance chemotherapy effect, therefore decreasing the drug dosage in the patient. In Canada, her work is an active collaboration with Dr. Alberto Delaidelli and Professor Poul Sorensen since 2023. So thank you so much, Jessica and Ferris, and we will pass it on to you.
Thank you so much for the presentation. Before I begin, I'm gonna give a little bit of like a short presentation on the tumor that we focus on, what is medulloblastoma, and especially why we're studying it. And that's before we start the lab tour. Please let me know if you have any questions. Don't have to wait up to the end. We can keep this as kind of like a conversation. I think sometimes that's easier to follow up. So just beginning a little bit of why medulloblastoma, why is that the tumor that we're targeting on this project. So between pediatrics, tumors, cancers, the most common one is those derived from blood, so leukemia, lymphomas. With the central nervous system tumors being the second in frequency, and in those kids, so from like 0 to 18 years old. And within those, the central nervous system tumors, the medulloblastoma is the most common. And I'm gonna bring it up the numbers a little later in the presentation, but it's also the highest cause of death in kids in cancer-related diseases. So medulloblastoma itself, by numbers, very recently, 2024, 70% of the patients are diagnosed are 10 years old or younger, and we have a high rate of high risk cases. So like a higher frequency of those high risk cases, especially in kids below 3 years old, which is one of the type of tumors that we're targeting in this project. 400 to 500 patients are diagnosed every year. This is US numbers, but we have similar frequencies both here in Canada and honestly mostly worldwide. And we have a 5-year survival rate of approximately 80%. So this 5-year survival, it's what we use in oncology to kind of quantified the risk of the tumor in matters of risk of death. And just to give, I don't know if you guys, I don't think you can see my pointer, but the medulloblastoma, they are located in the cerebellum, which is a part of the brain. It's located by the base of the skull and is responsible for developing speech, walking, eating, and other processes in the kid, which are very important for the development. Um, Medulloblastoma, like I said, there has like this average 80% survival rate, but we have different type of tumors into medulloblastoma, so like different types of medulloblastomas. And we have those who are low risk, it's called WNT, WNT, we pronounce it WNT. We have medium risk, and we have tumors of high risk, which is the group 3 medulloblastoma tumor. This is what we call high grade or high risk, and this is the type of tumor that we're studying, especially in our metabolism focus project here at BC Cancer as well. So now, why do we study medulloblastoma? This is one of the numbers that I wanted to bring it up to you. Although there are sometimes more common tumors in kids, the central nervous system tumors, they are the responsible for the highest rate of death in kids, and medulloblastoma is the most common of all. And group 3 tumors, again, we're gonna bring that up, that it's gonna end up picking that number. Cancer is still number one cause of death by disease in children, and the median age of death in those children, it's around 7 years, which is extremely young. I'm gonna talk a little bit about like what we have in curing treatment at the moment. Surgery, it happens most of the cases. It's a very, especially for medulloblastoma, it's a very aggressive type of surgery, which can again lead to a lot of side effects. That's something that it diminishes a lot of the life quality of the kid. Follow that by either chemo, radiotherapy, or a combination of both. Those again can also cause a lot of side effects in the kids following that. And by numbers, about 90% of those survivors for medulloblastoma have significant health issues because either the cancer itself or the toxicity of the treatments available today. We have in other type of tumors, what we call today like modern therapy, personalized medicine, and those include immunotherapy, like CAR T cells, and drug delivery systems that are more specific to the tumor, therefore causing less side effects in the kids. And then again, novel drugs who are less toxic to non-tumor cells, to healthy cells, but are way more effective, especially in cancer tumors. And those, always the focus is kind of lower the toxicity and increase both survival, but also the life quality of those kids while they're growing up. And how can science help? This is one of the parts that we've been doing. We have those cancers, we have specific markers on the outside of the cell that we can identify, and then we can target, so that the drug, the system, or the CAR T cells, which is kind of like immune system cells, they can identify specifically cancer cells and attack only those. So identifying those markers on the outside of the cells is what we have been focusing, and that the project that was awarded with the brain cancer tumor award. Before I pass it up to Faris, he's gonna explain a little bit more about these markers. I'm gonna just give a little bit of like an update on the metabolism part of the project that was introduced. So we work with one gene, and that gene was identified highly expressed on those his cases. So whenever the gene was more expressed, there was a decrease of survival in the patients. And by identifying that, we decided to see if it would be a good target for maybe a treatment for moving forward. And we saw that that gene can actually affect the cell metabolism drastically. And with that, we can try and make the cells more vulnerable to treatment, like chemoradiotherapy. And then again, the end goal is to make make as if we need less of the chemotherapy to cause the same amount of cell death, so the same effect on the kids. The genes that we've been studying, we've been getting some very good results. We see that it can, under specific nutrient deprivation, so specific nutrient modulation, we can actually make the tumor, impair the growth of the tumor, so like hold the tumor to a less extent. That can also help with the therapy. And we have been starting a project now working with inhibitors, so novel drugs. A lot of the drugs that we've been using, they are low toxic compounds, so usually inhibitors, natural derived inhibitors, that can also be eventually, if the results are promising, which they are so far, but be translated to a clinical treatment for those kids. So that's the part of the metabolism. Fars is going to talk a little bit more about this CAR T and immunotherapy.
Thanks, Jessica. So kind of my involvement in the project has been mainly through my co-op. So my time as an undergraduate student, I was hired by Paul to work with Alberto, and one of the main projects I focused on was this surfaceome project. So as described earlier, the surfaceome is essentially what's being exposed on the surface of a cell. Specifically proteins in our case, that's what we were interested in. And essentially the goal of the project was we wanted to compare using high throughput methods like proteomics. So the idea is we have technology where we can essentially prepare samples, isolate the surface membrane proteins, and then, essentially pass them into a machine that can tell us, it's almost like a computer in a way, but it tells us what proteins are detected in those samples. And so it was a great opportunity to work with like amazing technology that, I think otherwise I wouldn't have had access to, since it's pretty technical and expensive to process samples on using this method. But the idea is using mass spectrometry and proteomics, we found specific targets that are expressed on the surface of tumors derived from patients. And the goal was to see what things are expressed highly in these group 3 medulloblastoma cases, which are the high-risk ones that Jessica explained earlier, that are not expressed on normal human tissue. And the goal for this is once we find targets, and we found like around a list of 200 to 300 targets that were either not well characterized in the past in literature or have not been explored in terms of immunotherapies. And so now that we have this short list of 200 proteins, we're currently doing follow-up studies trying to pursue clinical tools that rely or that leverage these surface targets. And one of the things that Jessica kind of touched on is CAR T therapy, and that's kind of the thing that Alberto's currently studying. So the idea with CAR T therapy is you're essentially harnessing the immune system of the patient. So you're collecting T cells from the patient. You're then engineering them to express something we call a CAR, which is a chimeric antigen receptor. But the point is, this CAR, think of it as like I don't know what's a good analogy, but the point is it's going to specifically activate the T cell once it comes in contact with the antigen that it was engineered against. So in our case, we have these 200 proteins that are very promising hits. We are working on a short list of 4 to 5 proteins right now, since it's very difficult to focus on multiple ones at the same time. And so the point is we're going to design CARs that will specifically bind to these surface markers, and then hopefully carry out some experiments in vitro, which essentially means we're going to show you later like what in vitro cells look like, but the idea is we use cell line models where it's essentially a cell line derived from a cancer patient in the past, and it was engineered so that it can grow indefinitely in media. Well, not indefinitely, but for the most part, you can propagate it in media for a while. And so we can use those as experimental models instead of directly going into, patient cohorts, which is very, high risk, and in the past has been devastating since there's a lot of, non-specific effects that can happen with new therapies. And so, yeah, that's kind of the gist of what we're doing right now. The goal is to design immunotherapies that are more specific, less toxic, and less likely to target the normal human body. and will directly target the cancer. And since we actually examined a pretty big cohort of group 3 medulloblastoma patients, we're kind of optimistic that these targets are going to be broadly expressed across, a wide range of group 3 tumors. But, as science goes, you always kind of have to learn and adapt as you go. So we're currently still approving these first, 5 targets. And yeah, the work's still going on right now. If you want to start the tour.
Yeah. So, do you guys have any questions about this?
Oh, yeah.
Background, please feel free to ask us anything. I think this conversation goes. It's better if we have this. I'm gonna stop the sharing right now, so we can.
If anyone has questions, you can also add in the chat your question if that's easier for you.
Yeah. If not, we can start with the tour.
And feel free to ask during the tour too.
Yeah.
There's no, yeah.
So we're currently at the student room. I'm gonna start showing you this part. Is that okay if we start the tour?
Sorry, what did you ask, Jessica?
Is that okay if we start the
Oh
tour?
yeah, please, yes.
Okay, great.
Thank you.
So I'm gonna be changing between the front camera to the back camera on my tablet so you guys can see better. So right now with the student room, this is like an office for us. It's where we have our computers. That's where we do some discussion. We have the base material, some books, and some extra material for us to work with, and obviously some snacks sometimes, which is also important for us. And I'm gonna start the tour like I started from their lab. So we are multi-center, multi-lab center actually research. And so each floor has more than one professor. We call it PI, which is kind of like the main investigator. The lab access is blocked, so it's a safe environment for us to work. And also the data that we have here, which is sorry. Let me just one thing that I love about our lab, but I don't think I showed last week, is that the floor, it has those stripes, which are made to resemble the DNA strand. So like the double DNA strand. So and this is for us very interesting because we are a molecular biology lab. So it's just a fun detail. So those walls that I passed first, they are part of somebody like another PI. It's called Parisio, sorry. It's called Professor Parisio's lab. So I'm gonna start now with the part that is Professor Sorensen, and then I'm gonna show you some part of the bench and some of the main equipments that we use for this project. Let me know if if you cannot see or like the internet breaks, because the internet inside the tissue culture room and the microscopy room are not the best. So please let me know and I'll just wait a little bit until the connection is back. So here we start with the bench area. So bench is where we do most of like our experiments. There's always a lot of tubes, the pipetting, which is how we usually handle the samples. This is very safe, obviously gloves everywhere, and the lab coat so we can work on. There's several benches, and here at the lab, every student has one bench, so we can work in different projects and not have cross-contamination. We have more office area here at the back. This is one of the postdocs here at the lab. He's doing some research at the moment. We have, besides more bench areas, we have some fumes like this, which is for working with chemicals and dangerous products, mostly those who can kind of go up in the air. This is to keep it safe. It goes all the way up, and this is to filter whatever it's going out so that it's not exposed to the rest of the lab and it keep us safe. So there's glasses for safety, and we have a different procedure on working on those benches. We have additional lab office here. And we have this area here, which is for, sorry, can you give this to Leno?
Wait, sure.
So this is where we mostly work with protein. This time there is someone pipetting like a Western blot, so I'm just gonna ask her to tell a little bit what she's doing. If that's okay, just to add up a little bit. So this is Annalena. She's a PhD student here at the lab. She's working with Ewing sarcoma at the moment, sorry. And she's doing like a experiment which is called a Western blot. And what she's doing is that we have to add the proteins to the gel so we can run and separate the proteins and then eventually identify each one. And what she's doing is just pipetting in the gel, which if I could show you, it'd be really fun because you can see that we're adding like the blue liquid inside the gel, and we cannot see the gel, but we sometimes can see a little bit of how,
Wait, I can
the proteins are being added. Yeah, so that we can maybe see that. Yeah, like the blue lines being added to the gel. That's how we are working. Elena, what are you running now? Like, what are you identifying?
I'm currently running different conditions. So I have this inhibitor that I'm testing out right now, and I'm stressing cells and see if this inhibitor does anything to the protein expression in the end.
Yes. So yes, yeah, thank you so much. And I'm gonna continue with the tour. This is again mostly bench work for us. I'm gonna show you some of our materials here. This is more bench, and then we have some this is our chemical work. There we have a lot of materials that we need to work, so they keep it's everything for common use, which for me is very interesting in this lab. Sorry, I'm just it up again. Okay, so this is our bench area. This is where we do most of the experiments. I'm gonna show you documents that we have. They are shared with other labs, and because of that, we have documents that are in another PI's lab. So I'm gonna show you one now because, as I mentioned, we've been working with metabolism. And for us, one very essential equipment, it's called a seahorse. And what we do is that we can actually measure how the cells are breathing. So this is the equipment itself. It's called a seahorse. And it's mostly just like most analyzers that we see. We have one here which can count the cells. That's the Y one here. And I open like our results here just so I'm not sure if you guys can see it. But the different lines in this case, they show each cell line, and we can see by like how high or how low they are. Let me highlight one of those. Different like how the cells are breathing differently depending on what we do. In this case, we are comparing, like I said, the expression of a gene. So we've been working with total gene here, which is those lines here, half a gene expression here, the middle ones, and total knockout of the gene, which is the pink one. And for us, it's very interesting to see that we'll be able to modulate how cells are breathing, how cells are generating energy. And because of that, we can actually try and target those cell lines like in our favor in this case as maybe an additional treatment for the Asian patients. The next two rooms I'm going to show you is the microscopy room. which is interesting for us because it's also how we visualize the cells, both total cell number and also the fluorescence of the cells. I'm gonna show you some of the results as well. This is one of the rooms where the internet's not the best. So again, please let me know if I'm cutting too much and you cannot understand. Since this is the microscopy room, I'm gonna turn the light on first so we can see the machines. We have two microscopes. Those are fluorescence microscope. So they are very sensible. We can see, I'm gonna show you here that when we use the microscopy, we actually have a blue light happening over there that's going through the cells to try and show what we see here in this case, which is the cell nucleus in blue. And those green markers, there are, in this case, we're identifying lipids formation. We have this one, which is a treated one, that those lipids are being localized into specific points of the cell. And we have the untreated one, which is this one. You still see the blue one, which is the nucleus, but then you see the lipids all around the cells, right? And this for us was another, again, part of the metabolism study that we've been doing. So yeah, let me turn this off. Okay. And now I'm gonna show you our tissue culture room. This is where we keep the cancer cells for in vitro studies. So we have, this is the tissue culture room. We work on those BSCs. We have 3 of them. 1, 3. And those BSCs, they are biological safety chambers. And what they do is that they have parts that generate some airflow, and that airflow keeps whatever is inside protected from the air from the outside, and whatever is on the outside protected from the air on the inside. This allows us to work with those cells without contaminating them, and also safely work with them without contaminating the us and the work we're doing. Although the cell work, it's not dangerous or toxic for us. We also have more microscopes here. This is for normal visualization of the cells, not fluorescence. And I'm gonna show you here our cell. We have one with a camera, and I think I can show you the cells that we've been working with. I'm not sure if you guys can see, but I think so, yeah. So this is how we visualize the cells. This is cell enclosure. So those cells are currently being kept in a media that allows them to have like a normal metabolism in a sense. And then we can do the test and the treatment. Those are the cells lines over there. And the cells have to be kept in a very specific condition of nutrient, temperature, availability of CO2 and oxygen. And I think this is how we keep the cells alive. And the cells are kept because of that in a hood that is control for, like I said, temperature and CO2 levels. Okay. So this is mostly of the lab that we have here. We also do some work in all the labs in the building. So this is the 3rd floor. On the 11th floor, we have the flow cytometry, which we do a lot of the experiments. On the 7th floor, we have the proteomics. That's why we did the proteomics experiment that I was explaining to you before. On below the first ground, the first floor, we have the ARC, which is where we keep some animals for studies, some of the projects that we do. And yeah, so far, that's it. I wanted to show you guys, and I'm open for questions again.
Thank you so much, Jessica and Ferris. Does anyone have any questions? I see Ben has his hand up.
No, I was just giving applause because that was
Oh,
fantastic.
okay.
It was really, really amazing to see behind the scenes and all of the phenomenal technology that you're able to use to see everything at a cellular level. I'm amazed. Thank you.
Thanks, Ben.
Thank you.
Does anyone else
Yeah.
have any questions or comments?
Thanks so much, Jessica. I'll jump in, Julie. Great walkthrough. Fascinating. I agree, Ben. I'm curious from your perspective as in your position, what are you most excited about in terms of your findings and applying it to the care for the pediatric population?
So I think that we're most advanced in the metabolism project at this point. So I'm gonna talk about this one. We found a lot of interesting results, and what we do found, which for me is the most important, is that we be associating this gene with resistance on cisplatin treatment. So those cases that do not respond to treatment are actually have like a higher, again, a higher expression of this gene, and we're able to cancel that by knocking the gene out. And this is okay, this is something that is a lot of gene shows, but what for me is the most interesting about this one is that the gene is actually affected by nutrient deprivation. And what is that? So basically we took down the nutrients, see how the cells react, and we saw that it was very very intense results on that. And then additionally we tried taking one different nutrient at a time to see if there's one who will make a great effect. And we saw that if the cells didn't have glutamine, which is one of the nutrients for the cells, that gene was the knockout cells, the cells who didn't have, sorry, when I say knockout, it's the cells who don't have the expression of the gene, they were super sensible to that. And what's the importance for that? And we then kind of translated that to an animal study. And what we saw is that if we do like a simple 70% like diet, 70% calorie diet in the animals, we can also hold back the tumor growth. And this is without additional treatment. So those results for me, when it comes to the metabolism, this is for me is very interesting because we're talking about something that is easy and simple to do and has very impactful results. Obviously this is still animal. We always we get excited, but like still have to hold that. But for me, that's like, this is the results that we've been following up more intensively at the moment. We are continuing doing that. We're doing what we call a metabolomics experiment. So basically looking at the whole metabolism of the cell at once to try and see if we can target that, because there are like glutamine inhibitors that are used in clinical normally. And what we can do is just maybe add that to the treatment of the kids, have like a 4 or 5 times less cisplatin being introduced to a kid, and all because we could just modulate one simple thing. And for me, that's the most important part, when we can actually try and look at it from like the translation aspect and be like, where are we getting with this and why are we doing this and how important would that be, like everything working out. Obviously we still have a lot of work, but like everything working out, how important would that be that we can actually just take 5 times less of the very high, very toxic drug being introduced to the kids because of one simple modulation in one gene that we identified. For me, I think those are the results that make me come back to work every single day and continue the PhD.
Absolutely.
Yeah.
Absolutely. Thank you so much for sharing that, Jessica. That's amazing, especially that translation piece, right, to the impact of the patient and their family.
Yeah.
So key. I really appreciate you sharing that excitement. That's exciting for us to hear as well.
Yeah.
Thank you.
Does anyone else have any questions? Applause?
Thank you so much.
So Jessica and Ferris and your whole research team, your passion and energy really make a difference in the lives of families every day. We are thrilled to have been able to share with everyone the impact of your incredible work and dedication. And, you know, with our brain tumor community and our supporters, to see it come full circle and to be able to be a part of it. That is why we do what we do. And we are so appreciative of everyone. So if there's no more questions, I'm going to say thank you so much once again to everyone for attending today, and wish everyone a wonderful day. If there are any questions that you thought of later, please don't hesitate to connect with me, and I can pass it on to Jessica, etc. So it's we can keep that loop open. All right. So thank you very much, everyone, and have a wonderful day.
Thank you so much.
Thank you.
Bye.
Bye.